Well, hello everyone. My name is Michel. Thank you very much for joining us. I'm delighted to host Dr. Rick Helmich, and you might remember that we previously discussed the impact of stress and Parkinson's with him, and you can find the recording of that video, of that session on our YouTube channel. Today, we're not going to be talking specifically about stress, but we're going to explore the brain's backup plan, as we called it in the invitation. Dr. Helmich will show us how one part of the brain takes over when another part fails, which is fascinating. And we will learn why symptoms sometimes appear only when this backup system can no longer bridge the gap. Importantly, we will also discuss how to strengthen these pathways to keep the brain resilient. But before we start, like always, I would like to remind everyone that this session is for information and education purposes only. So if you are seeking medical advice, please make sure that you consult a medical professional. And as always, there will be time for questions at the end of this presentation. If you want to ask a question, nothing has changed. Please use the Q&A function at the bottom of your screen. And so for those of you who don't know us yet, No Silver Bullet stands for the idea that managing Parkinson's requires a multi-pronged holistic approach. And we really aim to share the expertise you need to build a holistic lifestyle that includes medication, obviously, but also exercise, relaxation, and technology. This, we believe, will help you to better manage your symptoms and slow the progression of the condition. We post the recordings of our webinars on YouTube, and you can find more details on our website. The website is nosilverbullet4pd.com, nosilverbullet number 4pd.com. But let's come back to today's topic, to Dr. Rick Helmich. Rick, thank you very much for agreeing to speak to us and to our community today. The stage is yours.
Thank you very much, Michel. Let me just start by sharing my screen. So today I will guide you through some research that we have done on the brain's backup plan, so how brain compensation plays a role in Parkinson's disease. And just a few words about myself. I'm a movement disorders neurologist in Nijmegen in the Netherlands. Where I see many people with tremor and also with Parkinson's disease. And I'm also a principal investigator at the Donners Institute, where I do research on the mechanisms of Parkinson's disease. And this is my team, so I'll be sharing a lot of their research with you today. So my research is focused on three topics, on brain compensation, which I will be speaking about today, and also on tremor. So what are the brain mechanisms that produce Parkinson's tremor, and stress, which is the topic that I spoke about the last time I was here. And what I really enjoy in my work is that as a neurologist, I am trained to really carefully look at people with a movement disorder, such as Parkinson's disease. But that in the research, I can use all kinds of techniques, such as brain scans, to really understand why certain things that I see in people with Parkinson's disease, why they occur. So to really understand the mechanisms. And I strongly believe that this leads to better treatments for people with Parkinson's. So you all know what Parkinson's disease looks like, but I just wanted to show a video of a woman with Parkinson's disease who has tremor and slowness of walking. And according to, most of the current knowledge, these symptoms are caused by the death of dopaminergic cells in the substantia nigra. And this leads to a reduction of dopamine in the striatum, which is a region deep inside the brain. And this leads to symptoms such as slowness and tremor and stiffness. And here you can see the the substantia nigra in the in the midbrain, and these cells in the substantia nigra project all the way up to the striatum and release dopamine there. And dopamine is very important for movement and also for thinking and for processing emotions. And in people with Parkinson's disease, these dopamine cells die, and that leads to a reduction of dopamine in the striatum. And that reduction can be visualized in certain scans. For example, this is a DOT scan. In panel A, you see a completely healthy person. In panel B, you see someone with Parkinson's disease in the early stages, and you can see that on the right side of the screen, the striatum loses its color a bit, meaning that the dopamine is depleted. And on the right side, you see more advanced stages of Parkinson's disease. And that is all true. But there's also a problem with this single explanation. And one reason is that the symptoms in Parkinson's disease start only when more than 50% of the dopamine cells are already gone. And that is surprising. So you could, of course, say, well, maybe we don't need all these dopamine cells, and maybe you can do perfectly fine with half dopamine cells before you get symptoms. But that is also not true. And evidence for that comes, for example, from animal studies, where animals are injected with a toxin that kills dopamine cells. And what they have found in this interesting study is that if you inject that toxin immediately, like the full dose in one go, these dopamine cells are damaged, and the animals get Parkinson symptoms such as stiffness or tremor. But if you slowly inject that toxin over the course of many weeks, even if it's the same amount, then the animal recovers. And that is what you can see here. I don't know, can you see my mouse, Michelle, on the screen?
Yes, we can.
Yes. So these light gray animals here that you see here, they were injected with the same amount of toxin as the other animals, but over a course of many weeks. And those animals, they recovered after a while. So, or actually, I should say it the other way around. So these light gray animals, they were injected with toxin immediately, and they remained stable over the course of many days. So meaning that they kept their motor symptoms, whereas the black dots, those animals were injected over the course of many weeks. They got the same amount of toxin, but they immediately recovered. So the lines all go down, and after a few weeks, these animals were healthy again. Even though both groups of animals, so the immediate dosers and the slowly progressive dosers, had the same number of lost dopamine cells. And that means that if a disease starts and progresses very slowly over the course of, well, in humans, that is many years, the brain can adapt and people can actually recover from that damage. Another reason why this explanation that dopamine loss in Parkinson's disease is the only thing that explains clinical symptoms is the fact that the progression of the disease over years does not correlate very well with the progression of dopamine loss. So here, these are complex figures again, but on the horizontal line, you can see different measurements that were taken in the same patients at baseline, then after around 12 months, after about 24 months, and then after 48 months. And you can see that the clinical symptoms, which are on the vertical axis, the UPDRS, they increase over time. But these increases in motor symptoms, they don't correlate very well with the increase of dopamine cell loss. So people, in other words, people who progress very fast can have the same reduction of dopaminergic cells, visible on DOT scans, as people who progress very slowly. So there must be other brain mechanisms that explain these different speeds of progression in Parkinson's disease. And finally, what post-mortem studies have shown, so these are studies that were done in people with Parkinson's disease who died of another cause. So, for example, who may have had a traffic accident or a severe heart attack or something else. And when they looked at the brains of these people with Parkinson's disease, and there are 12 people that they looked at. So these are every picture that you see, A, B, C, D, is a picture of a person with Parkinson's disease. And when they put them in order, so panel A here, this one is a healthy subject, and then panel B was a person with Parkinson's disease who had just 1 year of diagnosis. Panel C is someone with Parkinson's disease who had 3 years of diagnosis, 4 years, 5 years, etc. What you can see is that 1 year after the diagnosis, there are still a lot of dopamine cells. So the blacker the picture is, the more dopamine cells there are. After 3 years, there is still some dopamine cells, but after 4 years, the dopamine cells are completely gone. And after 5 years, 11 years, 14 years, nothing really changes in these dopaminergic areas anymore. Even though we know that Parkinson's disease, of course, doesn't stop after 3 or 4 years, people still get more and more symptoms over years, even after 4 years. So that means that, again, that dopamine is not the only problem in Parkinson's disease that causes symptoms. And what I would like to discuss with you today is brain compensation, which is which are brain areas that are not so much damaged by Parkinson's disease and that have the ability to take over certain functions. And the idea is that if people are better in compensating for the loss of dopamine in their brain, they progress more slowly and they have fewer symptoms in their daily lives. And this is not a completely new idea. So I don't know if anybody of you have seen the movie Awakenings in 1990. I love that movie. It was based on a true story written by Oliver Sacks. And it concerns people in a nursing home who had Parkinson's, not the normal Parkinson's disease, but Parkinson's because of a very severe infection. And that doctor in that nursing home discovers something. And I want to show you a part of that movie. I hope it plays well. I don't know if if the movie ran well for everyone at home, but what you can see is that this lady who has Parkinson's can only walk when there are visual cues on the floor. And that's what he found out. And you might, well, what is sad is that Robin Williams, who was the main actor in the movie,
actually got a Lewy body dementia himself, and also um, some of you might have seen in the news that Oliver Sacks might have invented some of his stories. So there might be questions whether this is actually a true phenomenon. But I often meet people who, with Parkinson's disease, who use these cues in their daily life. And this is one example of a patient from India who has Parkinson's disease, and he has severe freezing of gait. And his daughter, who is an artist, found out that when he walks the stairs, he can walk much better. And what she did is paint these fake stairs on the floor of his living room, allowing him to walk much better. So what you can, before the stairs painted on the floor, he has severe freezing of gait. But when he has this visual information, he can walk much better. There are also very personalized examples of people with Parkinson's disease who use brain compensation or compensatory mechanisms. For example, this young man who has also severe freezing, but who can play basketball. Or this man with Parkinson's disease and freezing, who is perfectly able to walk back, and also with freezing can walk much better. And can develop certain compensatory strategies to help them reduce their symptoms. So then the question is, what are these compensatory mechanisms? And before I proceed with that, it is important to realize what compensation actually is. And two criteria for brain compensation or plasticity have been put forward. And one is that it should be clear what is being compensated for. And in Parkinson's disease, that is the reduction of dopamine in the basal ganglia. And second, there should be evidence that the enhanced activation of a certain brain area, for example, is related to a beneficial effect on performance. So that when that brain activity is higher, people perform better. What is also initially triggered when it gets difficult. So when you do routine behavior, your brain might not be triggered to try and compensate. But when it becomes more difficult, when a task becomes more difficult, brain compensation is necessary. And there are some indications from neuroimaging studies, so from studies with brain scans, where these compensatory mechanisms might be. For example, this is a study from 5 years ago where they put together all the different studies that have been done in the last 10, 20 years, where people with Parkinson's disease were compared to healthy people. And they averaged all these brain scans and found that there are brain areas that are systematically not as active, so have lower activity in people with Parkinson's compared to healthy people. And these are the blue areas. And these blue areas, so for example, here in the middle, these blue areas that I'm circling now, they are the basal ganglia where there's a reduction of dopamine. So it does come as no surprise that this area is less active. However, what was very interesting is that there were also brain areas, and these are the yellow brain areas, where across many studies, there was increased activity in Parkinson's disease as compared to healthy people. So all of these yellow brain areas showed more brain activity during a motor task in people with Parkinson's than in healthy people. And that might be areas where the brain compensates. Now, to have a look at that further, we developed a task that people with Parkinson's disease performed in the fMRI scanner. And that was a task developed by Martin Johansson, a postdoc in my team. And there, people with Parkinson's disease were asked to press one of those 4 buttons while their brain was being scanned. And there were different conditions. So they could either, and those conditions were signaled by this, by the instructions on the screen. And those instructions were the 4 different circles that you see on the screen. And in an easy condition, a one-choice condition, there was only one gray circle, and people had to press that button with their finger. If there were 2-choice or 3-choice conditions, it was a little bit more difficult, and people had to select or choose themselves which of those 2 buttons to press. And those circles changed all the time. So it was not possible to always press the same button. And what we did, we asked 350 people with Parkinson's disease to come to the donors and be scanned in the fMRI scanner while performing that task. And we also had 60 healthy people who performed that task. So it was a very big group, which makes it a very reliable study. And what we found during this task is that in people with Parkinson's disease, when we compared that to healthy people, there was reduced activity in those blue areas. So the striatum again, so the part of the brain receiving dopamine projections, but also other areas such as the cerebellum and here the motor cortex. And I'll skip over these figures on the right. What was important, though, is that if we tested if the degree of activity in those areas was associated, so explained the clinical severity of the symptoms, that there was no such correlation. So, for example, here in the putamen, which is this comma-shaped area, there was no relation at all between the severity of the bradykinesia, so which is the stiffness and the slowness that people with Parkinson's disease had, and brain activity in that area, which is a bit weird if the bradykinesia is only caused by dopamine depletion in that area, right? On the other hand, what we did find is that people with more severe symptoms had lower activity in the parietal cortex and in the premotor cortex. So these are 2 areas that are on the outer surface of the brain, which we call the cortex, where there are no dopamine projections. So that means that people who had high activity in those cortical areas had less symptoms, less slowness, less stiffness than people who had very little activity in these cortical areas. And we found the same effect if we looked at cognitive function. So that is the speed of thinking. So people who had higher activity in those yellow areas, which are around the same area as on the left, were faster at thinking than people who had very little activity in these areas. We also saw that people with Parkinson's disease actually showed more activity in those cortical areas than healthy people. So it was not just that these areas were relatively preserved in Parkinson's disease. No, these areas worked harder, were more activated than in even healthy people. And that suggests that these cortical areas play a compensatory role in Parkinson's disease. And what I believe is that in Parkinson's disease, these cortical areas work harder because the deeper areas in the brain that receive dopamine are malfunctioning, are not working very well anymore. So then the next question that we investigated was, do these compensatory mechanisms then also explain why some people with the disease progress faster than other people? And what we tested was the idea that the speed of progression is not so much caused by the basal ganglia, dopamine depletion in the basal ganglia, because as I showed you at the beginning, already very early in the disease, this dopamine depletion is at its worst. It doesn't change a lot anymore after 4 years, but that it's actually the collapse of brain compensation that explains why people progress, why their symptoms increase. And well, what we did was we asked these patients that I just showed you, who performed that task, we asked them to come back after 2 years. And we measured their symptoms, which are on the vertical axis here, which are the motor symptoms, the UPDRS, which measures the stiffness, the bradykinesia, the slowness, the tremor. And we saw that they indeed, as you expect, they were worse after 2 years than they were 2 years ago. And that is, of course, what we know. Parkinson's disease changes over the years, becomes worse. And we also show that in these people that there is a loss of dopamine cells in the substantia nigra. And that is also not new. It is very interesting that we can show that with MRI scans, but that is also not new in people with Parkinson's disease. So the groups here on the right had more abnormal cells in the substantia nigra than people with than healthy people. But what is interesting is that we observed a correlation between the speed of progression, which is shown here on the horizontal axis, and the degree of brain compensation in the cortex, which is shown here on the vertical axis. So what that means is that people who progressed faster, so in whom the stiffness and the slowness was much worse after 2 years, were the ones that showed the most reduction in brain activity in the cortex. And those people who progressed very slowly, so in whom nothing really changed in those 2 years, still had intact brain activity in the cortex after 2 years. And that suggests that brain compensation in Parkinson's disease consists of increased activity in the cortex, and more specifically in 2 areas of the cortex, the parietal lobe and the premotor cortex, and that the collapse of this activity over time is associated with the increase of motor symptoms over time. So then the question is, of course, why does patient collapse? How can we preserve and protect
brain compensation in Parkinson progression in people with Parkinson's disease. And I don't have an answer yet to all of these questions, but that is one of the research areas that my group is working on at the moment. And I'll share some of the ideas with you. So why does brain compensation collapse? So one possible explanation is brain compensation collapses because the energy cells in the brain, the mitochondria, are not able to produce enough energy for these areas to be hyperactive. An important question is, how do we boost these compensatory mechanisms? How do we boost our mitochondria? And there's more and more evidence now that lifestyle, for example, exercise and physical activity, has an important role in improving mitochondria, and that also has an effect on the brain, such that the brain is better protected when the mitochondria, so the energy cells in the brain, work better. We recently wrote an article about that for the Lancet, which is about the role of lifestyle interventions in Parkinson's disease. For example, stress management, nutrition, physical activity. And what we believe is that these lifestyle interventions are very important for boosting the brain compensatory mechanisms in Parkinson's disease.
Rick, if I can interrupt you for a quick second, just to say, Rick, can you hear me? Just to say that actually Joanne will be talking to us in a couple of months about exactly that paper in the Lancet.
That's great. That is awesome. Yeah, Joanne is a great speaker, and yeah, we wrote this paper together, and Joanne will undoubtedly tell you more details about this paper. There are also, of course, pharmacological approaches to boosting brain energy, which is important for brain compensation. And another thing that we are working on in Nijmegen are non-invasive brain stimulation approaches. So in a study that we are setting up now, we have the goal to enhance these compensatory brain circuits in people with Parkinson's disease, which will hopefully lead to less symptoms in these people. So time flies. So I already have spoken for more than half an hour. I want to thank you very much for attending. I think some of the slides may have been difficult, but I hope you understood. And I want to also thank my team for helping me with all this research that we are doing. And I would be very happy to discuss or answer questions.
Well, Rick, thank you very much for the presentation. This is really fascinating. And actually, it's really interesting to see how this connects to our lifestyle. How can we improve our brain plasticity or give ourselves as many chances as we can to have parts of the brain bridging the gap with the other parts of the brain? And actually, you're touching to that topic, which Nocebo Bullet is based on, which is the idea that actually, aside from medications, which are critical, people should exercise, pay attention to their nutrition, and use the relaxation technique that you've mentioned before. So thank you very much for this. I'm going to the question. Actually, Suzanne is asking, when you measure the progress of Parkinson's in terms of evolution of symptoms, do you do that in periods when people are without medication? How does it work?
Yeah, that's a good question. So we did it in two ways. So
the gold standard to measure progression at the moment is to measure all the different symptoms, the tremor and the slowness in the hospital, which is the UPDRS. We measured those symptoms both without medication, so after 12 hours of no medication, and with medication. And both progressed over time, and both correlated also with the collapse of brain compensation. Of course, we know that this is not, well, sensitive technique to measure progression. So what we are also working on is, to measure progression better with wearable devices. For example, a smartwatch to measure the degree of tremor or the degree of slowness.
Thank you very much. Actually, interesting question from Thomas, who is asking if it's possible to use EEG instead of an fMRI to probe the progression of PD and the cortical activity.
Yeah, that's a great question. So I haven't used EEG to measure progression. Measure very well with EEG are rhythms. So the rhythmic activity of the brain. What you cannot measure with EEG is everything that lies under the cortex. So you cannot measure the basal ganglia, you cannot measure the substantia nigra. So the big advantage of functional MRI is that you can measure both the deep areas and the more superficial areas at the same time. So that is why we, in my research group, use MRI.
Thank you very much. Someone is asking whether macrodosing, so I'm not pronouncing it right, I know the mushroom psilocybin or LSD, does it help increase compensatory mechanism for people with Parkinson's? Has someone looked at this?
That's a very interesting question. So I know that there's a lot of interest in these type of substances because they can, well, increase flexibility in some way. But I don't think that has been shown in good studies yet in Parkinson's disease. So, but it's an active research field. Yeah.
Excellent. Maybe for one of your PhD students.
Yes, maybe.
Lisa is asking whether she's talking about neurofeedback and says that neurofeedback addresses the compensatory cortical areas by training up underpowered areas or reduce over hyperconnectivity. And so how can we move the whole scientific field more into this area?
Yeah, that is also a great question. So neurofeedback, so for those of you who don't know what neurofeedback is that neurofeedback is that you measure brain signals and you give that information back to the person in real time. So that you can see, for example, by some shape on the screen that becomes larger when your brain is more active or smaller when your brain is not as active, so that you can see in real time how active your brain is. Because if it works, you can teach people in the scanner to boost their own brain activity, so to increase their cortical activity, for example. But it's also very difficult because, of course, you need to be able to measure that in real time, which is very difficult, technically very difficult. And it might not work for everyone because if you move too much, for example, or if for all kinds of reasons, the brain signal is very noisy, you cannot, it doesn't work. And the other learn to do that, and that takes many, many weeks to be able to learn how to influence your own brain. And all that time, you need to be in the scanner because you need to see that brain activity. So institute colleagues of mine are doing neurofeedback in healthy people. actually to reduce stress so that people recognize on the screen when their stress system is too high, and they can reduce it using relaxation techniques. But we haven't that in Parkinson's disease yet, simply because it's very complex.
Thank you very much, Rick. Two questions here from Mark and from Sarah that I think are touching to the same topic. Mark is asking whether longer-term levodopa use and its side effect could it be responsible for the collapse of the brain compensation? So too much levodopa for too long. And Sarah is asking, do your results suggest anything about taking dopamine after 4 years? So did you look at the impact of people taking dopamine for long term and the negative impact it might have on the ability of the brain to bridge the gap?
Yeah, that's an important question. So the short answer is no. I don't think that levodopa, after many years, does any harm to the brain. I don't think levodopa can damage brain compensation. It's actually the other way around. I think that the combination of dopamine which is important for the deep areas in the brain, combined with compensatory techniques, which involve the superficial layer of the brain, the cortex, that this combination can be important, that it can be synergistic.
Yes.
It's also, I guess, also important to stress that I also think that levodopa continues to play a very important role, not only in the first 4 years, but also after that. So we know that levodopa remains highly effective also in people with later stages of Parkinson's disease. You might wonder, how does that fit with the picture I showed you, that after 4 years the dopamine cells are completely gone? Well, that's easy, because after 4 years your own dopamine production might be gone, but those cells that receive dopamine, so the receptors, are still there. So levodopa continues to play an important role in Parkinson's disease. But in the later stages, brain compensation plays an additive role.
And I guess that actually dopamine helps us also exercise better than without dopamine. So it's an indirect impact on, if you want, the factors that can help the plasticity.
Exactly. And it also reduces stress in some people and anxiety. So I think levodopa is still very, very important.
So I have a question here from someone who is asking, what nutritional advice would you give which might be important in this case? I'm personally thinking reducing oxidation, reducing inflammation, maybe, I don't know. We talk a lot about mind diet or Mediterranean diet. What would you say would be helpful in this case?
Yeah. So what we describe in that paper that Joanne Trin will also speak about is that the best evidence there is currently is that the Mediterranean diet is beneficial for Parkinson's. That is probably caused by a few things. One is that it contains a lot of fibers, and because vegetables contain fibers, and fibers are very important for promoting motility of the gut. And that motility is very important for absorbing not only the medication, but also other nutrients. So fibers are important. And the second thing is that a Mediterranean diet indeed reduces inflammation. And inflammation, we know that in people with Parkinson's disease, inflammation is a little bit increased. Not as much as if you have the flu or if you are sick, but just a little bit increased. And that inflammation, we think that contributes to the continuous damage to brain cells. So if you can reduce that a little bit, that might be beneficial.
Thank you very much, Rick. Someone has an interesting question here. Mark is asking, what are the implications of your research for deep brain stimulation? I don't know, I'm reading between the lines, but actually, would it open the door towards stimulating other parts of the brain that facilitate or sponsor more neuroplasticity from them, or to allow them to be bridging the gap for longer, for instance?
Yeah, yeah. Thanks for that question. So, well, we are doing this study at the moment, or we are setting it up in Nijmegen to try and stimulate these cortical areas. The brain well is particularly suited for small areas because you stick a small electrode into the brain and you can stimulate, well, a few millimeters. So it is very effective when a very small area in the brain is malfunctioning. And that is the subthalamic nucleus in Parkinson's disease. So there it works perfectly well. For the cortex, the brain stimulation is less suitable because the cortex is much larger. So it's a much larger area that you want to stimulate.
Yes.
So there we need other techniques. For example, stimulation of the brain via coils on the skull.
Yes. That basically gets me to a question by Pilar. She's asking if you're considering low intensity ultrasound as a non-invasive treatment for just that kind of situation.
Yes, exactly.
Or is that, yes, it is.
Yeah, yeah. So that is the technique we are using in Nijmegen. So for so focus or HIFU new technique in which ultrasound beams from many areas are sent into the brain, and in one particular area, for example, the thalamus or the subthalamic nucleus, the temperature is increased, causing causing very small lesions or holes in the brain that can help as treatment. There is now also non-invasive sound, which that is sent into the brain is not as high, and where there are no lesions being made in the brain, but the brain activity is increased or decreased. And that is indeed to try and, well, manipulate brain activity without damaging the brain.
Wait, the line is not great, so I switched off my camera, hoping it would make a positive difference. I'm not sure it does, but you sometimes break up. I mean, it is not so bad that it doesn't work. It's just on the borderline. But so I'll switch off my camera. We'll see if that helps. Jody has a question about the fact that people with Parkinson's are very heterogeneous, so they have a different mix of symptoms. And she's asking whether this is perhaps compensatory mechanism that vary by individuals as well, that would lead to a variety of symptoms.
Yeah, yeah. I also think that is the case. So I think that the degree to which brain compensation works in Parkinson's disease differs a lot between people. And why that is the case, I don't know. It might be that in some forms of Parkinson's disease, the cortex is early on already damaged by the disease itself, so that it cannot compensate. So that could be one reason. Yeah.
Someone is asking actually, it's interesting because this is something I do every day, and I know it's good for everyone. Are there any mental exercises, such as crossword puzzles and word games, that are known to strengthen the parts of the cortex that help compensate? So as we exercise the rest of the body, should we also exercise our brains through simple crosswords or other ways of staying basically awake and alive?
Yeah, I don't know about crossword puzzles. I think everything that challenges your brain that keeps you active helps. People sometimes tell me that when they had a lot of stress or they were overburdened in one way or another, and that they had more symptoms than before. And what I think might go on is if you have a lot of things going on, you have to attend a lot of things. And your brain is basically overburdened. You cannot focus anymore. Those compensatory mechanisms are wiped away. So you need some focus in order to use these cortical compensatory mechanisms. So yeah, and that ties this story that I told you today to the stress story that I told you earlier, where things like mindfulness are important to increase the focus. And that might help because it strengthens or it allows brain compensation to take place.
Julia has two questions. And actually, the first one is that would you consider most compensatory mechanisms to be based on distraction? Or does this not explain the mechanism enough?
Yeah, it depends what you mean with distraction. So if you look at these areas that are hyperactive in Parkinson's disease, which are the parietal cortex and the premotor cortex, that these are areas that are important for attention, for attentional focusing. So I think uh, not because they are distracted, but rather the opposite, that they can focus their attention on the task they are doing.
Yes, great. And the second question she had is quite technical, but let's go for it. Have you measured the size of the amygdala in patients with PD? And it is, she mentions, in general, smaller with people such as people who suffer from PTSD. So have you looked at that specific aspect?
So I personally haven't looked at that, but it is an important a brain nucleus that is important for anxiety and for stress. And we know that people with Parkinson's disease have more anxiety and more stress than the average population. And in normal in Parkinson's disease. I have looked at that, but I would expect it to be. Yeah.
Thank you very much, Rick. We have quite a few questions. Actually, I will stop in 5 minutes, so I'm so sorry. We won't be able to answer everything. I'm just trying to pick a few. Steve was asking whether you believe that ketogenic diets or fasting can have an impact on neuroplasticity.
So I think the connection was a bit unstable there. So ketogenic diet, right?
And fasting. And does it have an impact on neuroplasticity? I know we have had Dr. Matthew Phillips a couple of times talking to us about the small study that he did on using keto for people with Parkinson's. It was encouraging, but a very small sample. And that this is his darling topic. If anyone wants to know more about that, you can look at our YouTube channel. You will find tons of things. Well, actually, at least 2 sessions by Matthew Phillips. But I don't know, actually, Rick, if you have an opinion on this.
I don't think that has been investigated. So intermittent fasting or ketogenic diet, I don't know. I'm sorry.
No problem. This is actually very fair to say. And I realize the range of questions here is quite wide. Yeah, I have a couple of questions, actually. Again, it may be unfair to ask you, but it's still something which is very much on our radar screen, which is the red light or the photobiomodulation, red light or other types of lights. Have you looked at how this can impact potentially the stimulation of certain parts of the brain that can play a part in neuroplasticity?
we haven't yet looked at that. So I do think that visual processing plays a role in Parkinson's. So, of course, we know that people compensate by using visual cues for example, this staircase that was painted on the floor. So visual processing must play a role in these compensatory mechanisms, same as rhythms and sounds. If red light or if certain visual stimulation techniques can actually increase compensation, I don't know. I don't think these studies have been done yet.
So the question here, it's actually a question I had myself. The person is asking, is multitasking a good way of improving cerebral cortex functioning? And actually the twist I want to put on that is that I have, in my routine, which is on our YouTube channel, quite detailed, I have started this year to go for more variations, to try things I have never done before, to meditate in virtual reality, to try again rollerblading. If you don't hear from me in a few weeks, that will be me falling in the lake. But basically trying to do things that are different or difficult, like, for instance, boxing, and not only the strength it requires, but the mental aspect of it, of remembering complex sequences of movements. So do you think that actually, whether it is multitasking or keeping your brain traveling, for instance, seeing new things, do you think this may help the cerebral cortex function better, or it can help the health of the brain in general to be mildly challenged, if you want, within reason?
Yeah, that's a great question. So actually, you're saying two things, right? You're saying multitasking, which is doing two things at the same time, and you're saying exploring new things. So learning new skills and learning, new challenges. And to start with the last, I think that learning new things is a great way to stimulate your cortex. So what we know, if people learn new things, is that you first start by activating your cortex to learn it. And when it becomes a routine, then those memories are brought down to the deeper parts of your brain. So I think learning new things is a great way to stimulate your cortex. Doing two things at the same time can be challenging because then you might overburden your cortex because it has to switch constantly from one thing to the other.
It's also a source of stress for many of us.
Yep. Yeah, exactly. Yeah.
So I think we're going to have time for one last question, and this is a question from Julia, which actually, yes, there are two questions from Julia. I'll take the first one. She mentioned that a new study in Nature that you have seen about SCAN, basically, actually, I think that I'm sure you have a list on that because it was quite an important article. And actually, they were talking in that article about overconnectivity, disrupting not only movement, but also thinking. And do you want to say a few words about that and how this could correlate to what we've discussed today?
Yeah, so that's a great question. So the SCAN is indeed a relatively new network that binds together different parts. So not only the motor network, but also cognitive or thinking network. And what that paper in Nature showed was that people with Parkinson's disease indeed have hyperconnectivity between their basal ganglia and this SCAN network. I think before we make very firm conclusions about SCAN, it needs to be replicated by other groups. We've looked at SCAN and saw if we tried to see if that SCAN network was the same as this compensatory network that I showed you, and that was not the case. So I think they are two different things: brain compensation and deficits in the SCAN network. But it is indeed highly relevant and interesting to see how they interconnect. So I think that will be done in the future.
Perfect. Rick, thank you so much. Actually, someone was asking whether the presentation would be made available. Would you be able to share your screen with me? Unless there is anything that patients you don't want to show or... So
Yes.
I'll let you think about it. If you feel comfortable with it, you can let me know tomorrow, or remove any slides that would show identifiable patients, for instance.
Yeah, that's fine.
Perfect. Well, look, Rick, thank you so much. We've reached the end of our time today, and I must say that it was an incredibly insightful presentation. We had a large number of people. We had great questions, as you could hear. I don't know how many times you said great question, and I think that reflects the involvement of our audience. And a recording of the session will be available either tomorrow or the next day. And our next webinar will take place in a week's time, on Thursday next week, the 26th, for the second installment of our Food is Medicine series. And we will focus on the gut-brain axis. We often hear that the gut is the second brain, but what does it actually mean for the daily management of our condition from a nutritional standpoint? So I will be looking forward to joining you again, or for you to join me on the 26th. And Rick, I wanted to say thank you very much for this. This was really amazing. Thank you not only for the time you're spending with us today, but also for your dedication to finding ways to help our condition in making your career out of it. So thank you so much for everything.
Thank you, Michelle, for the invite and for organizing this great platform. Thank you.
It's a pleasure. Thank you so much, Rick. Bye-bye.
Bye-bye.